Sciences for funding this visit. But I'd also like to thank my many friends and colleagues here in Nanjing, particularly Professor Rong, Guan Bin, and Zan Ren Bin, who can't be with us. He's in Canada at the moment. But all of these people have put a lot of work into actually organising this visit and making things happen. When I was asked to give this series of lectures, I thought I'd try and put together sort of 4 snapshots of different parts of the geological record. Some parts I know very well, other parts I don't know quite so well, but it's quite important to sort of build up the story by actually starting with some basic principles and starting a little bit with the Precambrian before next time we're going to talk about the origin of the Metazoans and then the Cambrian explosion, the Ordovician radiation, and then finally talk about what these actual events, how they have fashioned, how they have affected the structure of Phanerozoic life. So what I'm going to do today is essentially start with some basic principles. A lot of these things you probably know already, but in order to build up the whole story, I think it's important to start at the bottom and start with some sort of basic ideas, some basic principles. So I think if we can put the lights down a little bit and see what's going on the screen. I think as paleontologists, we have a lot of very important challenges. And I'll just put up some sort of very simple statements at the moment for you to actually think about. And the first one is that we're now fairly certain that life originated a long time ago on the planet. It originated probably about 4 billion years ago. Today, if you look around the world, and biologists are not agreed about the actual figures, but a lot of estimates fall between 10 and 20 million living species we have today. Some people say less, some people say a lot more. I've seen estimates of 50 million. But there's a lot of living species today on our planet. However, if we go back through the fossil record, sort of conservative estimates for the numbers of fossil species are around 500,000. That's the sort of figure that you see a lot in the literature. So obviously there's something very wrong in the system if we have 10 to 20 million living species and we only have 500,000 fossil species. Well, as you all know, part of the reason is it's relatively rarely that a fossil is actually preserved. So we're very lucky to actually find fossils because they've gone through so many processes to actually become a fossil. So in fact, we're missing a huge amount of basic information in the fossil record. And this is one of the points I'll continue to emphasize, is that one of the key sort of areas of our science is the actual study, the taxonomy of fossils, and it's so essential to get out there and find more fossils and keep collecting fossils and keep describing fossils to build up our knowledge of the fossil record. Another point that's quite interesting, and I read somewhere recently, that the estimates of all species that ever lived, 99% of them are probably extinct. Okay, it's something that's very difficult to prove or falsify, but it's an interesting thought to bear in mind. The point I'm going to cover today is that really for about 3.5 billion years of Earth history, life was microbial. We had fairly stable microbial type communities that dominated the Earth. Nothing sophisticated, nothing complex, like the sorts of communities we have today. Life supposedly was fairly simple for a very, very long time. And the sort of animal evolution we're talking about, the evolution of the metazoans, that whole process seems to have occurred fairly recently. Okay, there are different estimates from molecular biologists and from paleontologists, but a very rough figure of when animal evolution or metazoan evolution began to really accelerate was probably during the last 600 million years. So really, if we want to put the whole thing in a nutshell or try and summarize what we're trying to do, I think our biggest problem as paleontologists is to try and understand why we have this picture here, the early Earth, darkness, no atmosphere, still bombarded by meteorites, still bombarded by ultraviolet light, a very miserable, inhospitable planet way back at the start of the Precambrian, to a very different planet that we have today, that has tremendous biodiversity, tremendously complex communities that have developed throughout geological time. And as I said before, one of our challenges through examining the fossil record is to try and see how much we can understand about this entire process. So I think it's really exciting to be a paleontologist and get really deep into this type of study to see what we can develop. What I want to do is, in the structure of this lecture today, is to talk a little bit about the way that we understand the structure of life, the sorts of tools or the sorts of frameworks that we need to develop to see the actual fossil record and understand it. So that will be the first part of the talk. The second part, I just want to say a little bit about the early attempts to develop what we call the tree of life. And this is very fundamental to the way that we understand evolution through time. And again, fairly briefly, but a very significant, important part of this talk is just to emphasize the importance of the fossil record. Because really, when it comes down to it, it is our fossils that really tell us the most about evolution of life on this planet. And then I want to talk a bit about the Precambrian and a bit about this rather strange microbial world. Well, it's probably not very strange. I think the time that we're living in at the moment is probably rather strange, because microbial world lasted, as I said, for about 3.5 billion years, and that was life on Earth. And finally, I want to hop up to the top of the Precambrian and into a very different type of ecosystem, and a very important and significant ecosystem, which is the Ediacara ecosystem, which in a way is a transition between the microbial world and the metazoan world, dominated world, that we see today. So these are the sort of 5 areas I'm going to look at this morning, and in a way they'll link into what we're going to do later with metazoan evolution, diversifications, and the structure of Mesozoic ecosystems. Okay, well, a lot of this sort of stuff, understanding the structure of life, is pretty basic stuff that you've covered before, but probably I'll try and give you a slightly different angle on the whole process. I think that as paleontologists, the most significant breakthrough that ever has been made is that by Nicolaus Steno. And he built on the work of the Greek philosophers and various other people, that he actually recognized that these guys here, which are called tungstones, and they occur very commonly in the tertiary rocks of the Apennines in Italy. Steno was lucky enough to be down on the coast in Tuscany when the fishermen brought in a huge dead shark. Now, Nicolaus Steno, or Niels Steensen, he was originally Danish. He had studied medicine in Leiden in Holland. He made a lot of discoveries about the actual anatomy, human anatomy already. And he moved into geology when he moved to Florence. And in studying geology in Florence, he developed a lot of very interesting ideas. So when he was sitting there on the beach and he saw this shark, and he looked at the head of the shark, he thought, well, there's a remarkable similarity between the teeth of this living shark and these tongue stones that are found as fossils. Well, he dissected the shark very carefully, and I think he proved without doubt that these were actually fossils. They were the remains of once living animals. They didn't actually grow within the sediments, as many people thought. They were actually the remains of a former world. So I think this was a very fundamental advance in our thinking, the actual recognition that these things were actually fossils. But the way wasn't easy, because although Robert Hooke and others in Great Britain already were already recognizing fossil wood and fossil shells, there were a number of hoaxes that caused us a lot of problems. And one of the most famous was the Professor Beringer of Würzburg in the early 1700s, and he was given these remarkable things by local collectors. And of course he thought, wow, these are absolutely incredible. And he started describing these things as fossils. And one of my Swedish colleagues has been fortunate enough to go and look at the original specimens, and they're quite remarkable. They're absolutely incredible. Now, these things were very quickly shown to be hoaxes. Okay, people had just chipped these out of the rock. And okay, they made Professor Beringer look extremely foolish, but unfortunately for many years they made all paleontologists look rather foolish, because as often happens in science, if one person makes a mistake, then everybody is credited with that mistake, and the whole area of paleontology took a while to overcome these hoaxes and go forward again with some real paleontology. So, fossils were recognized in the 1600s. They'd been recognized before by the Greeks, but now they were very much on the map. The second thing I want to mention, because it's a thing that's also very important, is the whole idea of stratigraphy. So even if we recognize fossils, we've got to put them in the right order. So we need a stratigraphy, and this is a very important part of our work. And actually, you may think it's rather strange that I go back to the Italian Renaissance, to Leonardo da Vinci, who made many inventions and many great discoveries and many great paintings, such as the Mona Lisa and the Last Supper and all these sorts of things. But he's also credited with the fact that he rediscovered sort of perspective of three dimensions in his drawings. And this is a famous sketch that's in the Uffizi Gallery in Florence, in which you can demonstrate that Da Vinci had sort of discovered this three-dimensional landscape perspective. And American colleague Gary Rosenberg has written quite a lot on this, and it's quite an interesting topic. So with this perspective, it becomes possible to actually look at landscapes in three dimensions and actually see the structure of rocks. Now, this was very important for, again, Nicolaus Steno. Something went wrong, I think, with Steno. Later in his career, he became a priest, he became extremely religious, and he died very young in poverty. But nevertheless, perhaps his greatest scientific discovery was this idea, the law of superposition of strata. And nowadays we say, well, it's very obvious that the oldest rocks are on the bottom, the youngest rocks are on the top. But back in the 1600s, it wasn't so obvious. And he was working again in the Apennines. We've got Florence in the background, and here he is looking at some cross-bedded strata in the foreground. But his idea was this principle of superposition, that if you have a rock succession, the old rocks are on the bottom, and then younger and younger rocks are deposited on the top. So again, that was a very major breakthrough. Unfortunately, we had no real idea about geological time, how long it actually takes. And okay, there's many estimates, that people have put, from biblical estimates of 7 days to more precise estimates of thousands of years. But it was actually this rather strange-looking gentleman here, James Hutton, who was a scientist that developed a lot of his ideas during what we call the Scottish Age of Enlightenment in the late 1700s, when there was a lot of philosophers, a lot of scientists, the philosopher David Hume, for example, the economist Adam Smith, all these people. It was a centre of learning, a centre of knowledge. And James Hutton was pursuing his studies in terms of geology, trying to work out how the Earth actually functioned. And he recognised what we now know as unconformities. Okay, everybody knows what unconformities are, that's not a big problem. But he went around Scotland and he found 3 major, what he called unconformities. And this is the most famous one at Sicker Point, just outside Edinburgh. And here you see vertical strata of Silurian age, and here you see sort of shallowly dipping strata of Devonian age, okay, old red sandstone on Silurian turbidite. And there's a gap of about 30 to 40 million years between these 2 sets of strata. And James Hutton sort of reasoned that there must have been a lot of time. These strata must have been deposited, they must have been folded, tectonized, uplifted, eroded, and then this new set of strata was actually deposited on top. And this gave Hutton the idea of deep time, that the Earth had been around for a very long time, and sort of geological processes operate in deep time over many, many millions of years. So he came up with this famous phrase that this demonstrated to him that the Earth showed no vestige of a beginning, no prospect of an end. So it was a very long, endless process that we're looking at. So this was the start of giving us a little bit of time to our stratigraphy, and this was sort of followed up by a number of studies that began to sort of crystallize our understanding of stratigraphy, our rock framework. Giovanni Arduino, again in the Apennines, developed this model in which you had a primary basement, a secondary set of strata, here mostly Mesozoic, and then poorly consolidated molasse here. So this was his primary, secondary, and tertiary systems. Okay, the primary and secondary have gone. The tertiary is still hanging on, just at the moment. It's not quite gone yet. It may stay, it may not. The whole idea was that he could recognize this in the Apennines, and around this time many other people recognized these divisions everywhere else in Europe. But the problem is they weren't actually the same age. They could recognize a basement and a cover and unconsolidated sediments on the top. But in fact the basement, for example, in Scotland might be Precambrian in age. In the Apennines it's Variscan, and the cover sequences might in fact be Lower Paleozoic. So although this structure was recognized, it was clear that they were of different ages. So this is a problem. So, just add the work of William Smith, who was a canal builder, an English canal builder, and he was an engineer. And he recognized in his traverses from London over here to Wales that there was a definite order. He knew the law of superposition. He had an idea of Arduino's primary, secondary, and tertiary. But he also was able to look in a lot more detail at the individual fossils that were found en route. And so, for example, up in the chalk, there were rather nice echinoids, ammonites, those sorts of things. But by the time you got down to Wales, into the Cambrian, for example, you were dealing with trilobites and brachiopods and those sorts of things. So he had developed his own system of actually correlating these different strata by fossils. Okay, it was very crude, but it was, again, another fundamental advance in the way that we were thinking. Well, around this whole time, other geologists throughout Europe mainly were recognizing geological systems and geological periods because they had particular types of fossils. And so by the late 1800s, most of the geological systems, I think all of these geological systems, had actually been defined around different parts of Europe. Now, the very big problem with the geological systems is that they were defined more or less on breaks or on gaps in the succession, because European geologists at that time, Adam Sedgwick, Roderick Murchison, even Charles Lapworth, these sorts of people, thought that if there was a break, if there was a gap in sedimentation, an unconformity, then that marked the division between two different systems. And that's quite logical, okay, if you think in that particular way. Where it began to break down was that when people looked at other continents, went to North America, South China, those sorts of places, they found that in those particular places there weren't gaps, there was continuous sedimentation. So the actual whole methodology of setting up this whole group of systems was in a way rather false. It worked for Europe, but it didn't work anywhere else. And if you come to correlate bases of systems on gaps, you're actually correlating nothing with something else, so it's not really going to work. So it was essential that the International Commission of Stratigraphy develop a whole new and rigorous way of actually building up this geological timescale, or this geological column, that's based on defining strata types in sections of continuous sedimentation where they have good fossil control from a number of different groups, so that you're actually correlating rock with areas out with that strata type section. And I've just been very fortunate to go and look at the first boundary strata type that was actually defined for the Ordovician system in Zhejiang province, and this was largely due to the work of Professor Chen and his colleagues here. There's a rather fine monument here of the key fossil, Ostrodentatus. And the important point is that if you look through the sort of boundary strata type section, you have continuous sedimentation. You have shales rich in graptolites, you have calcareous units that are rich in conatomes. You have a continuous section that you can actually correlate the base of the Darwinian stage with other strata throughout the world. And in fact, in China, for our own system, the Ordovician, there are in fact 3 of the 7 international stages are actually defined here, which I think is a considerable credit to not only the excellence of the sections and the very good fossil control, but also the very hard work that's been done by our Chinese colleagues and colleagues from abroad in actually getting such excellent data to be used in these strata type sections. Okay, so that's the structure. Okay, so we have a stratigraphy, and that's a really important thing if we're going to go any further. But we've also got to have a sort of tree of life. We've got to try and understand how a lot of this fossil data hangs together. And this, I guess, is one of the first trees of life. And next year, there's going to be a frenzy about Charles Darwin. I think most paleontologists and biologists in some way will be celebrating the 200th anniversary of his birthday. But his Origin of Species, which was published back in 1859 and was a sort of bestseller at that particular time. They printed many, many editions of it. It sold extremely well. Strangely enough, he only has one figure in the whole of the book, and this is the one figure. And it's a very simple figure, and if you read the text, this is actually the Cambrian here. And he had a very great problem with the Cambrian, because at that time virtually nothing was known about the Precambrian. And so it appeared to him that life started at the Cambrian. But of course, Darwin was an extremely clever guy, and he realized that there must be roots to a lot of these lineages way back into the Precambrian. There must be a rich Precambrian life prior to the base of the Cambrian. But his figure here shows a couple of very interesting things. It firstly shows this whole idea of speciation through time, these splitting lineages, with life becoming more and more diverse as we go through time. The second thing it shows, which is quite interesting, is some sort of gaps here. And he was very aware of the concept of extinction events. And so it wasn't just one simple progression of speciation and diversification. There were, in fact, times when a lot of things were extinct. He also talks a lot in the text about the increasing complexity of life. And so we start with fairly simple organisms at the bottom and end up with fairly complex organisms at the top. Well, many historians have managed to discuss this tree in very great detail and have made a lot of interpretations over some of the fine lines. But I think for me, the main points are this missing Precambrian record, the increasing biodiversity and complexity, and also the implications of these gaps and extinction events. So there's quite a lot in this very simple diagram. Darwin was also influenced a lot by this geologist here, Charles Lyell. And if you were studying geology during the 1800s and even into the early 1900s in many parts of Europe, this would have been the textbook that you would actually use, Charles Lyell's Principles of Geology. And this was first published in the early 1830s. But it is a fantastic book, and there's all sorts of very interesting information. But one of the key points within Charles Lyell's book is this idea of what he calls gradualism. And Lyell was very much in favour that geological processes took place at a very slow and gradual rate throughout geological time. And I think this very much influenced Charles Darwin with his idea of evolution. The evolutionary events were fairly gradual. And this is a diagram from Charles Lyell's book. It's probably one of the first sort of attempts at statistical paleontology. And he tried to recognize the different series at that time within the tertiary. The oldest one here, the Eocene, recognized at that time, going up through the Miocene, the older Pliocene, and the newer Pliocene. He tried to recognize these things statistically, which was quite interesting. So what he did was that he tried to look at the sort of ratio of those animals that are still living today and those animals that are actually extinct. So if you come up to the present day, the ratio would be 100%. It would be all the animals are actually living. Okay? And that makes a lot of sense. But as you go back and you look at perhaps mollusk species and you look at mammalian species, there's more and more and more of the actual animals he's looking at have become extinct before the present. And so if you go right back, then there's only a relatively few of these species are actually around today. And so on the basis of these ratios, he was able to actually define statistically these different stages. Well, it probably doesn't work very well in practice, but it was an attempt based on rates of extinction to actually define geological stages. Well, in the last number of years, we've had a lot of very important studies of particular case histories in the fossil record to try and see whether speciation does in fact occur gradually, or perhaps it occurs very quickly and then nothing happens for a few million years. And some of the most rigorous studies include Peter Sheldon's sort of study in the late 1980s, in which he looked at a lot of trilobite species through an interval within the mid-Ordovician. And what he was able to do along a stream section in Wales was to collect literally hundreds of specimens of all these particular taxa of trilobite. And he measured certain key characters. I think it's the number of ribs on the pygidium of these different trilobite taxa, and he tracked them through geological time, getting younger and younger and younger. And what he did was, and here you've got the number of ribs on the bottom and geological time on the side, was he plotted the taxa as at these particular levels in the strata. And here, if we look at, let's look at Abigia carella here, which is a very well-known distinctive trilobite. And here, we've got it starting here and maintaining and getting fewer and fewer ribs, and then taking off again, and then sort of getting fewer, and then moving off in this direction with more and more ribs, and then going back on itself and going up like that. Well, two main things came out of this research, in Peter Sheldon's view, was that it was a gradual process. Okay, ribs were added to the pygidium, or ribs were subtracted to the pygidium. Okay, it's a fairly simple basics of the study, but things took place gradually. The second conclusion, which has been known before but never really formalized, was that you can get evolutionary reversals. Okay, so not only does the trilobite gain ribs, but it actually starts to lose ribs as well. So these were two quite interesting and important conclusions based on this study. Now, there's been a number of other studies, different fossil groups, showing that in fact you get this sort of what we call phyletic gradualism. But in the early 70s, Stephen Jay Gould and others developed a rather different model, that the model of punctuated equilibria, that in fact evolution doesn't take place at a steady rate, as Darwin and Lyell and others had thought. But basically what you see is that species don't change for many millions of years. Nothing really much happens to them. And then there's a sudden burst of activity. And then they go back to this period of what we call stasis. Now, Peter Williamson, working in the Turkana Basin, actually did one of the best, most rigorous studies, and he looked at a lot of different lineages, not only of these gastropod mollusks, but also some bivalve mollusks. And he showed that there were intervals here in which you saw this very sort of sudden jump in morphological change. And this, to him, and Stephen Jay Gould and others, was definitive evidence that there was punctuated equilibrium. Now, there's a lot of issues about sampling and gaps in the record and those sorts of things, and how you assess the morphological characters. So it's not a very simple process. But at the moment, I think nobody has really decided if there is one model or the other. In some environments with some taxa, evolution proceeds slowly in the sort of phyletic gradualism mode. But in other types of environments, evolution can sort of hop along very quickly, and then nothing much happens in the punctuated equilibrium. So it seems to depend on a lot of variables, and there's no one mode, I think, is the definitive way that evolution works. Well, in developing a sort of tree, an evolutionary tree, I think we have to go back to the work of the great German zoologist Ernst Haeckel, who put together, I think, one of the the earliest and most comprehensive trees of life, and he published it as an actual tree. He was a sort of very interesting scientist, because later in his career he got very much involved with the interface between art and science, and published a number of books, a number of monographs, sort of showing fossils as objects of art. And unfortunately, this didn't actually do his reputation any good, because he had actually done a lot of very fundamental and important work, which is recognized today. So this is his sort of tree of life, and you can follow some of the branches with some of the most primitive unicellular type organisms down at the bottom, to, predictably enough, gorillas and orangutans and gibbons and chimpanzees and man right at the top here. But this would have been based on morphological data in developing this sort of tree of life. Well, things are a lot more complicated now that we have such good and reliable molecular data. And this is one, I'm sorry that this is sort of in Danish, it's for something else, but this sort of shows the complexity now of the whole system, of the real importance of a lot of the types of bacteria. And we, sort of as animals, find ourselves out here on the very close to fungi, based on molecular data. And a lot of these sort of more primitive, simple sorts of organisms are extremely abundant, extremely common, and they must have formed the basis for this microbial world in the Precambrian. And if we can make it even more complicated and look at some of the more recent trees by Patrick Keeling and his group, life is extremely complicated at a small scale. And we as animals really form an extremely small part of the whole system. And, well, here we are down here, very close to the fungi again, the animals here. But there are literally many, many groups of very important microorganisms that are quite different from each other, that we really don't come across as paleontologists. But if we focus just on animal evolution or metazoan evolution itself, there's been a lot of work done recently, principally by Kevin Peterson's lab at Dartmouth, and he bases a lot of his work on microRNA, and his group have been building up a series of microRNA trees, which are giving us a much better structure to actually understanding the evolution of the metazoans. And interestingly enough, these trees show that some of the sponge groups are in fact polyphyletic here, but these are fairly relatively simple organisms, the cnidarians here, and then we come into the sort of main groups of the metazoans, with the deuterostomes here, with the echinoderms, hemichordates and chordates here, and then the protostomes down here. One of the big problems is still that there are, in fact, polycotomies on his tree that are not very easy to resolve. So there are groups that look to us to be very different, such as the mollusks and the brachiopods and the annelids. But in terms of microRNA and his molecular studies, these are very hard to resolve. But nevertheless, we have a very clear structure of the tree of life as it involves the main metazoan groups. Okay, so we're coming a bit of a way up through the process. We do actually have a geological structure to work with. We have our stratigraphy, and that's pretty good. And we are developing a tree of life that paleontologists are involved with. We know the structure of evolution of that tree of life. why are fossils so important? Well, I've just put together some pictures of fossils, and it's just a random selection, and there's no reason why I've chosen any of these particular fossils, other than they were on my laptop at the time, and they come from Denmark and Greenland in this case. But I just make a very general point, again, that I made in the introduction, that without this sort of data, without actually collecting these fossils and studying them and doing taxonomic work, we can't really go any further with these rather nice macroevolutionary studies that I think a lot of us want to do and be excited about. And so this sea urchin from the chalk of Denmark is beautifully preserved, but you know, we need to know what it is. We need to study it in great detail before we can do some of the exciting things of understanding its ecology and understanding how it actually lived in this rather strange